Swayam Prabha. Digital India. Educated India.
Welcome back to my course Industrial Biotechnology. Now in the last lecture I try to tell you the production of high fructose corn syrup, which is I told you mostly used by in the Western country. Now I am little bit coming to a special area that is called metal leaching. How biotechnological application we have in the metal leaching, and lot of industry they are using the metal leaching process for the recovery of metals. And this metal leaching process basically that is applicable where the ore contains very low percentage of metal, because as you know that when we use the ore high concentration of metal, then we go for the chemical process. We go for chemical, that is very that is well known, but you know that when the ore contains very low amount of the metal, then we go for this metal leaching process. Leaching basically that signifies something you are liquefying. So now question come how this leaching take place. Now you know that when you talk about any kind of ores, that in metal ores particularly, it is kind of iron sulfide. You know, iron sulfide is S-2. This is the sulfide. You know, I want to say this is a sulfide, and this when it undergo this leaching process, this converted sulfate. So when it converted to sulfate, this is insoluble, and this is soluble. So this is so the metal sulfide, this is converted to metal sulfate. This is how the solubilization take place, and this is called the metal leaching. So the bioleaching or metal leaching is a simple and effective technology for metal extraction from low grade ore and minerals concentrated by the use of microorganisms. So I shall discuss very interesting that I can discuss that you know how this is taking place. The microbes such as bacteria and fungi convert the metal compounds into their water soluble form, are biocatalyst of their leaching process. Now this is two organisms very interesting, that thiobacillus thiooxidans and thiobacillus ferrooxidans. There is a very commonly used microorganism for the bioleaching process, because largely available in the metal dump, you know, where the ore has been dumped. If we isolate the organism from there, we can find these two organisms commonly present in this. They are largely used for solubilization of sulfide. So it looks like this. You can say that you have a kind of rod shape, the thiobacillus thiooxidans. It is also kind of rod shape, and thiobacillus ferrooxidans, that also rod shape. This is always found to be present in the leaching dump. Now if you look at the history, this is the copper recovery from mine water in the Mediterranean areas that take place 3000 years ago. So this is a very old process, and the role of bacteria in bioleaching was shown in 1947. 1950s, the copper dump leaching that has been carried out. In the 60s, the first industrial copper heap leaching operation also take place. And the first industrial gold bioleaching plant was established in 1980. Nowadays, 40 plants in industries use copper, gold, zinc, cobalt, and uranium. So this lot of industry, they use this process for recovery of the metals, and particularly when we talk about the high costly metals like uranium and gold. This is, I will show you that at the end that how the different process affects the economy of the process, economy of this metal recovery process. So the feature of the organism involved, it is single-celled organism and chemosynthetic metabolism that you have and derived from carbon dioxide, oxygen from the atmosphere and required acidic pH. So acidic pH is mandatory, they required in this for this organism. Now if you look at the bacteria helps in regenerating major old oxidizer, mostly the ferric ion. This reaction takes place in the cell membrane of bacteria. Like I was talking about the sulfide FeH2 and ferric ion, then water, it will form the ferrous ion, and this is the thiosulfate and 6 hydrogen ion. And then this is the reaction, this sulfur also, this is ferrous ion is converted to ferric, and this sulfur converted to H2SO4. And this thiosulfate on presence of oxygen is produces sulfate, and here also direct reaction we can have this how this sulfite is converted to sulfate. So when this I can give the example of the ore like chalcopyrite. Pyrites is largely available in as a ore. So this is sulfite ore, and then when you treat with this organism and when bioleaching takes place, it produces sulfate. So it will come, all the metals will come in the soluble form. Now if it comes in the soluble form, then your recovery process will be very simple, and through the electroplating process or different process we can use for the recovery of the metals. So we have two type of leaching, bioleaching process. One is direct bioleaching, another is indirect bioleaching. In the direct bioleaching, the minerals which are susceptible to oxidation undergo direct enzymatic attack by the microorganism. So this is directly metal that this here the microorganism directly attack the metal ion, and it produce the converted to sulfate. Indirect means bioleaching, the minerals bacteria produce strong oxidizing agent. It produces some kind of ferrous is converted to ferric, and this ferric goes inside the cell and then help for this solubilization of the protein. That in indirect method, the bioleaching that of minerals bacteria produces strong oxidizing agent which react with metal and extract them from the ore. This is the natural occurring bioleaching process is very slow. It is not very fast. For commercial extraction of metal by bioleaching process is optimized by controlling the pH, temperature, humidity, oxygen and carbon dioxide concentration. So this is usually done in presence of oxygen, and there are three type of leaching process we have. One is called dump leaching, another is heap leaching and vat leaching. Now, if you look at the cost analysis of these three processes are different. That in case of dump leaching, this is usually executed at the mines itself. At the mine we have all the ores. So there we have some collection basin. We put we sprinkle the microorganisms. Then we collect this soluble material, recycle back to the system again and again until unless most of the material has been solubilized. So this is a low costly process. The reason is that this is executed in the mine itself. And heap leaching also usually done at the mine, but it is done in a planned manner. So they have more provision of aeration, more provision of that organized manner. So monetary involvement in this heap leaching will be comparatively high. But vat leaching we usually take place in a control vessel where we can maintain the temperature, pH and all these things. So your rate of reaction and time of reaction obviously will be very high. So this is the three things we have. We have dump leaching, we have heap leaching, we have vat leaching. Now dump leaching Generally size of particle is high, and ores is processed for many years by sprinkling the acid water in the dump site. Dump site means I want to mean the mines. The leach solution percolates through the dump and is collected the ditches at the base of the dump. The initial plant of copper recovery by dump leaching from oxide ore were successful and lead to the development of heap leaching of ore for higher grades. Now, if you look at the heap leaching, it is, as I told you, it is a little bit organized manner. We use that, we can collect, they have the provision for collecting the leaching material. We can do the concentration here. This is the collection basin that we have, and this is the processing plant we have. From this we can recover the metals. We can say this is the uranium recovery. That is kind of a example that has been given here. In heap leaching, ore is arranged in heaps. This is the heaps. This is, you can see, in different layers. Formation is there. The aqueous solution containing the microorganism works on the heap of the ore. So this is the sprinkler. We sprinkle the acid solution with the microorganism on the ore. So when it travels through this, then sulfide will be converted to sulfate, and it is solubilized, and liquid will comes out like this and produce the leach liquor. The leach liquor is used for metal recovery. Now, vat leaching, as I told you, vat leach unit is a rectangular container. This is usually done in a controlled environment, drum, barrels, tanks, or vats. Vats usually very big and made of wood or concrete or lined with material resistance to the leaching media, equipped with agitator. The treated ore usually coats, and leaching reagent may be added to the leaching object in different ways. So this is a costly process. The vat leaching is basically it is a costly process, and here you can have the control environment, and we already pointed out that microorganisms, they are very sensitive to the environment. If you maintain the environmental conditions properly, then your microorganism can work very fast. So that plays very important role. But this is comparatively with the dump and the heap leaching, this is comparatively costly, but metal recovery will be very high in this process. Now, copper leaching, we have different type of ores. I told you that the chalcocite, chalcopyrites, and covelites. So that this is the typical copper ore that is available in the mines. Because in India, this copper leaching process largely used by the industry. So for the recovery of copper. Now, copper leaching is operated simple heap leaching and in situ leaching process. In situ means that this plant is taking place at the mine itself, and dilute sulfuric acid is percolated through the piles, and liquid coming out from the bottom of the piles rich in minerals, and liquid is collected and transported to precipitation plants. Metal is precipitated and purified, and this is the reactions that we have. Copper sulfide, the Cu2s converted to Cus, and Cos is converted to this copper, and copper is removed. Like a final reaction is this ferrous oxide and copper and ferrous ion that we have, and iron is transferred to the oxidized pond and converted to ferric. This is the reaction how it is take place, and ferric ion produces as the oxidation of ore. It is pumped back to the piles, and sulfuric acid is added to maintain the pH. The gold and silver leaching, the microbial leaching of refractory process metal ores to enhance the gold and silver recovery is one of the promising applications. The gold is obtained through the bioleaching, the arsenopyrites or pyrites. Silver is also obtained from bioleaching, the arsenopyrites, but it is most readily soluble than gold during the microbial leaching in iron sulfide. So uranium leaching, uranium extracted when insoluble tetravalent urium is oxidized with hot sulfuric acid and ferrous sulfate solution makes a hexavalent urium sulfate. The pH required for this reaction is 1.5 to 3.5. Temperature we maintain 35 degrees centigrade. The reaction is like this. The uranium oxide, they react with ferrous sulfate, and this is ferric sulfate, and then it produces the uranium sulfate. This is in the soluble form. So uranium leaching is the indirect process where the ferroxidants that is involved for the uranium extraction, and they do not directly attack the ore but iron oxidant. The pyrites reaction, they initially produce iron. This is the kind of reaction that we have, how the sulfide convert to sulfate. Now the main factors that affect the bioleaching process here physicochemical is temperature, pH to keep the ferric oxygen reaction, and affects the leaching rate, microbial composition and activity. That the activity of the organism plays very important role, needs to be low to obtain the fastening the leaching rate, to keep the ferric ion and metal in solution. Electron acceptor needed to be chemical and biological oxidation. Now main factors of the microbial things also very important. Microbial diversity, culture and population density, and the metal tolerance, that plays very important role. The mixed culture tends to be more robust and effective than pure, because if 2-3 cultures present in the ore, then what will happen that if one particular organism is inhibited, but other organism may not be inhibited by the same concentration of the metal ion, so it may work. So we always find that in case of metal leaching process, it is quite effective. Here I want to point out that particularly in case of I shall discuss about cheese making industry, and the cheese making process, one important step is the card making. In the card making process we required Lactobacillus casei and Streptococcus lactis, the different type of organism, and this organism, they have the bacteriophage contamination problem, and due to this contamination problem, what is happening is, one, if a bacteriophage is very specific for a particular bacteria, if it attack a bacteria, that bacteria will be inactivated. So that is why if you use the mixed culture, then always it is favored the card formation. Is similar to here, that always mixed culture or microbial diversity culture always help in the metal extraction process. Now population density, high population density tends to increase the leaching process, because this is the organism, they secrete the enzyme and that take part in the reaction. Naturally the metal, that population density plays very important role. Metal tolerance, as I told you, that concentration of metal as when it is in soluble form, then your organism will not be inhibited. But as soon as the metal coming in the soluble form, the metal ion concentration increases, that may give some kind of inhibition effect on the microorganism. So the metal tolerance also very important in this particularly bioleaching process. Now that other factors like minerals composition, particle size, surface area, porosity and presence of other metal sulfide, this is very important. Composition provides the electron donor and trace element. The particle size, more surface will be available for contacting with the bacteria, and more leaching will be more. That the surface area, the particle size and surface area, they are quite correlated with each other. Porosity, crack, pores or particles give rise to the internal area, that is also very important. Presence of water metal sulfide, that minerals having the lowest potential is generally oxidized fast. So benefit of bioleaching is the simple and inexpensive, employed for collecting the metals from waste or drainage, and extract refines an expensive metal which is not possible by other chemical process. no poisonous sulfur dioxide emission take place as in smelter, no need for high pressure and temperature, ideal low grade ore, solid ore can be used, and environmental friendly process. The disadvantage of this process is several. It is a time consuming, take place 6 to 24 hours or longer, have a very low yield of minerals, required large open area for treatment, maybe no process control, and high risk of contamination since it is a open that if you look at dump and heap leaching, it is an open process. So the contamination problem will be very high, inconsistent yield because bacteria cannot grow uniformly. This is at the end I want to discuss that how this this process affects the that costing of the process. That we have two examples with us with the copper recovery system and the gold recovery system. We will make a comparative study with respect to dump leaching, heap leaching, and you have hat leaching. And I told you, if you look at this process, dump leaching operational cost will be very less, heap leaching will be comparatively little bit high, and vat leaching will be very high. So it is like this. Now when you do it here, they did kind of economic analysis. Now if the reservoir capacity is 50 metric ton, it contains 1% copper, its annual capacity of production is 3 to 8 metric ton, they found it is economic. Now even if it is the reservoir capacity is 250 metric ton, the average grade is 0.5% copper, then the annual production is 12 to 22 metric tons, then they found is economic. Now here you see that the heap leaching also 75 metric tons and 150 metric tons, 1% copper, they even 5 to 9, 6 to 12 we find economic. But vat leaching with the same, when you use the 150 and metric tons and 1% that copper and 6 to 12 metric ton here, but it is not economic. Why it is not economic? Because the operating cost, but cost of operation of the process vat leaching is comparatively high as compared to heap leaching and dump leaching. So that is why this is not economically feasible. So that depends. Operational cost plays very important role in the metal leaching process. Now I have another example with the gold leaching process. Now we find that if the reservoir capacity is 10 metric tons, then we have average grade of gold is 3 gram gold per ton, and annual production is 0.5 to 0.9 metric ton. It find non-economic. Even in vat leaching it is not economic. Then 10 metric tons, if the concentration increases 4 gram gold per ton, it is 0.5 to 0.7 metric ton, it is find marginal economic. But economic things will come when the gold concentration comes to 5 gram gold per ton and annual production is 0.5 to 1 metric ton, then it finds that your vat leaching process is economic. Now if you come here in case of this heap leaching process, that even 10 metric tons and 3 gram the gold per ton is present there, and annual production is 0.5 to 0.9 metric ton of gold, we find it has the marginal economic. So we find that metal leaching process is quite interesting. The reason is that this is applicable when ore contains very low amount of metal, because this metal recovery process through the chemical process is no good, because this usually take place at high temperature and high pressure. So it is very costly process, and that is why when your ore contains very low amount of metal, then your bioleaching process will be mostly effective. And we find that two organisms, that is Thiobacillus peroxidans, this is the organism which is very effective for the leaching process, and where sulphide basically converted to sulphate. sulfide is the insoluble material that remains in the ore, and this is converted to sulfate. When it is converted to sulfate, the metal will come in the soluble form. Then you can precipitate out the metal and get the pure metal from that solution. So the recovery process will be a little bit easier. Only the problem is that when you go for this process, we find this reaction is very slow reaction, and also concentration of metal ion in the solution also quite less. So that is the only bottlenecks is this. Still we find that there are 40 different industries we have throughout the year, and they are largely operating and for this metal recovery process. So I think this is very useful. This is a very good application of the biotechnology, that biotechnological science for the recovery of some useful metal for the human benefit. Thank you very much.